Uh, Firstly, thank you so much for coming onto the podcast. This is super exciting to have you on.
My pleasure.
And I should say, massive congratulations on the Nobel Prize this year. That's hopefully one day I can follow in your footsteps and
Thank you.
discover new types of RNAs. What's something like totally unexpected about the Nobel Prize process that they don't tell you? Do you have to like require to learn how to ballroom dance or something?
Oh, yeah. Um, well, I think the part that's been particularly surprising for me is the reaction of people around me, at my institution, family members, friends of family members, will congratulate family members, right? That, about this. So it feels like people kind of take a sort of celebratory ownership in the Nobel Prize if when it happens near them, you know. So UMass, at UMass, I felt a very surprising, degree of even outpouring of congratulations, but also shared joy, to be associated with, this event. So folks at UMass, I feel like are really um, enjoying this fact that we got this Nobel Prize, much more than I expected at all. So I feel like that was really educational, and it means that its significance is much broader than just me. It's about society, I think, celebrating science, and sort of reaffirming kind of people's enthusiasm for science. If you're already enthusiastic, it's a lot of good vibes. Let's say that's what was surprising for me.
Good vibe is always good. It's yeah. I know one quote you mentioned was, we're all sort of in this together. Science is a very big collective effort, and that definitely yeah resonates with what you're saying. So diving into microRNAs,
Mhm.
I know the paper came out in 1993, if I'm correct.
Yeah.
Could you could you walk us through the moment you realized you discovered something as groundbreaking as microRNAs? I mean, of course, that was quite a while ago, and when you discover something new, you don't really sort of realize down the road how significant it's going to be. But was there some sort of aha moment?
Yeah, that's definitely was a kind of a progressive process, right? To come to, let's say, an aha moment. If I think if I was to point to a moment when when it dawned on me that microRNAs were, let's say, really important, um, in a broad sense, was when Gary Ruvkin's paper came out in the year 2000, where his lab had shown that LET7, the second microRNA that was discovered in C. elegans in the Ruvkin lab, um, he showed that that microRNA was conserved in sequence, across all the bilaterian animals. And so that meant, when I read that Nature paper, and I didn't see it coming, right? So the paper was new to me. That finding was new to me, and so I realized, holy cow, that means that Lit7 and Lin4 that we had discovered in C. elegans were just an example of a class of regulatory RNAs that was evolutionary ancient, and so our common ancestor of all these animals had these microRNAs, and so that meant there must be many, many more of them, um, and that they are doing, important regulatory functions in all the animals, not just C. elegans. So that was really a key watershed moment for me.
Also, when I was trying to come up with questions for this podcast, I was like, my background is also in like the RNA-seq side of thing. So I want to dive bit more into the technical side, but I also want to keep it a bit more general. So
Hmm.
trying to balance the two and maybe ask one or two philosophical questions. But just to keep that balance, for sort of the general audience out there, what are microRNAs? I know this is changing gears a little, but would, love to hear it from from you.
Yeah, yeah, that's a very important question, because the micro sounds like they're small, and that really was one of the distinctive features of microRNAs is that it's a RNAs, as you know, are the products of genes. That's how the genes are copied. So the information in the DNA can go out and do its thing. And typically, the average, let's say, gene or the typical gene is, is copied into a messenger RNA. And that messenger RNA is long enough so that it can be decoded or translated into a protein polymer. And, those are pretty long. And so your typical messenger RNA would be at about 1,000 nucleotides long, right? And so now there are shorter RNAs that people, of course, knew about, transfer RNAs that are aiding in the translation step where that mRNA is copied into proteins. Those are whatever, 70 or 80 nucleotides long. And so when we, ended up finding this first microRNA, which was the product of this gene Lin4, in C. elegans, um, we weren't looking for anything peculiar. I mean, we were really expecting that this gene, again, Lin4 would code a protein because that's typically what genes do. And, we knew the Lin4 was carrying out some really interesting regulatory activity, because when you delete the gene, the animal experienced profound disruptions in development, and essentially the kinds of disruptions were developmental timing defects, right? So this gene was of great interest just based on the fact that the mutation caused such interesting developmental phenotypes. But again, there was no reason to expect that these interesting phenotypes necessarily needed to reflect some kind of novel type of regulator, not at all, right? And so when we found that it was a small RNA, we said, okay, this is really interesting. This is a kind of a new way for, or, a previously undescribed way for one gene to be regulating another gene, because it makes a little RNA of only 22 nucleotides long, but that's enough information for that RNA to specifically base pair, with its target messenger RNA. And so, um, yeah, so that type of small RNA was again discovered by Gary Ruvkun when they went ahead and found the gene product for this other gene, LET7, and LET7 was an interesting gene again because it had developmental timing defects. So again, about a 21 to 20 nucleotide RNA. And so for a while, those RNAs were called small temporal RNAs or sTRNAs because we thought, well, they have to do with timing, right? In C. elegans. But that name got discarded when other microRNAs were found that were not involved in developmental timing. They were doing other sorts of activities, um, you know, be it in development or regulating physiology or so on. So then everybody got named microRNA.
So we know that the discovery of microRNAs over the years has unlocked, exciting therapeutic possibilities from microRNA like nanoformulations for cancer immunotherapy to like microRNA-based PROTACs for targeted protein degradation. Nothing, despite setbacks like off-target effects and some halted clinical trials, there is a lot of promise in both the therapeutics and diagnostic side of things. Um, Among the current developments, which near-term applications in microRNA therapy are you most excited about? Now, this might also be a super broad question because the whole therapeutic space is very large.
Yeah.
But keen to hear some of your thoughts there.
Yeah, this is an area that I'm not, this is one of the many areas that I'm not an expert in, right? So it's a huge area. There's lots of publications, of course, where folks have sought to find microRNAs that would be useful in diagnostics or prognostics, right? For example, in cancer, the idea being that the kinds of microRNAs that would be found, let's say, in circulation in a person's blood plasma could be indicative of the presence of tumor of some type. And I think there's been some, fruitful developments in that area. Um, the other side is, when the microRNA has understood to play a role in tumorigenesis, either because its absence is helping the tumor to grow. Those would be microRNAs that are normally functioning as tumor suppressors. In miR34 and LET7 are examples of those. I know there's been a lot of work trying to bring those to bear against tumors. And I guess the challenge in most of these cases is trying to deliver the microRNA specifically to the tumor, because the microRNA has roles, normal roles in other tissues. So you would really want to do it delivered specifically. And um, I think this goes for a related kind of therapeutic effort where one uses an siRNA, which of course is closely related to microRNAs, because they're also 22 nucleotides long and they're associated with argonaute and so forth. So, again, the delivery issue is a major one. And I think it just, the bottom line is it takes a long time and a lot of effort and a lot of clever innovation to really engineer biological systems so that you can deliver, your favorite therapeutic directly to the cell types you're trying to, where it needs to act. Now, of course, knocking down microRNAs that are oncogenic is also, of interest, and there's been a lot of work in this area as well using oligonucleotides. But again, the delivery problem is there. So that would be my summary: is that I'm most excited, let's say, about the folks who are really working on this delivery issue, trying to deliver, small oligonucleotides, whether they be RNAs or anti-RNAs, microRNAs or siRNAs or anti-microRNAs, specifically to tumor cell types, where they need to do their work, whether that's the bulk tumor or the metastases, the cell, the stem cells in the tumor. Um, I think that, obviously, advancements in that area is really going to ramify across the board for lots of diseases.
And what I really like about that area, the delivery side of things, it's such an interdisciplinary science as well, because you have biophysicists working on it, you've got bioinformaticians working on it. Because in this topic, I mean, in this area, I saw a paper where they use DNA origami to make like nano cages where you can sort of make a little microRNA capsule there. And then there was another paper that looked at molecular dynamics side of things. And then, of course, these days with a lot of language models being trained on sequencing data and all of that coming together just shows like these are sort of like five different fields in a way,
Yeah.
tackling one problem. And I think that will sort of accelerate the advancement and
Yeah.
development of these therapeutics over the next few years as well.
Yeah, I think there's also another dimension that crops up a lot. And again, I'm not involved in this, directly involved in this field of trying to engineer therapeutics of any kind. But as a member of the faculty of UMass Chan Medical School, I sit on the committees, the thesis committees and, qualifying exams and so forth of students, many of whom are involved directly in this, effort. They're therapeutics engineers in training, working in labs that are developing, nucleic acid therapies here in the RNA Therapeutics Institute, the RTI, which is a big department here at UMass Chan. So what I've learned from being on those committees is that the challenge, more often than not, almost always, is the specific delivery, efficient delivery, and managing efficacy of the oligonucleotide or RNA once it gets into the cell. That can involve somehow getting it into the cytoplasm, from the endosome. And all these are problems which are challenging because they bring you right face to face, let's say, with the biology of the cell and the unknowns about the biology of cells and the unknowns about the biology of the systems, tissues, and the organism. Why does this agent go to those cell types and not those cell types? And why in a tumor does the agent go into the normal cells that have infiltrated it, but not the tumor cells, and what that means is there's biology there that we don't understand because if we understood it, we could just turn some knobs and make it go in, right? And so I think the other dimension, and you're right, it's a very interdisciplinary dimension, which is to try to take, when we run up against a problem in therapeutic development and delivery in particular, could we bring into the conversation folks who would be curious as, let's say, cell biologists, right, or biophysicists, curious about trying to understand the why behind it not working. The idea being that if we could understand why it's not working in the way we intend, we'll learn something new about fundamental biology. Because think about it this way, if we knew enough about fundamental biology, we could just engineer the sucker to go right in, And since we can't, that means we're ignorant of a lot of fundamental cell biology. And so if you're interested in basic science questions of trying to uncover new principles of biology, one way would be to try to follow the trail of failed therapeutics, right? In the same way that we follow the trail of mutations, right, that cause disease. And these tell us, hey, there's a gene, it's important, and we could learn some new biology from it. So I think that's a long-winded way of saying I think there's opportunities, to learn new biology, but also the biology would then enable folks to be, have a streamlined pathway to engineer some of these things, especially in the context of platform technologies where you're using conjugates to deliver to particular cells, and you're using a nucleic acid to target a particular gene. If we can solve the fundamentals on both those arms, let's say, of the strategy, that would be really exciting.
Fundamental science, that is usually always the answer. Let's start there. I think I got my PhD project sorted as well. Amazing. So there's a few more questions I wanted to ask, but I am acknowledging the time left. But I think I might keep this as the final question. It was really, really inspiring, witnessing the scientific partnership you've shared with your partner, co-authoring so many fundamental papers that reflect a great collaboration and dedication to scientific discovery. And I remember you've highlighted that, and I quote, "The success of science and of the individual scientists is derived precisely from the fact that we do it together." Given this, collaborative spirit and recognizing how traditional structures often emphasize the principal investigator or senior author, what are your thoughts on the Nobel Prize ruling on limiting the number of awardees? In terms of this rule can sometimes seem at odds with the collaborative nature of modern research, and maybe sometimes out of step with the reality of shared discovery. And more broadly, how do you think the recognition systems like the Nobel Prize could evolve to better reflect the collaborative and often gendered imbalanced nature of today's scientific community?
Very good question. Right, and the tone of your question, I mean, one subtext of your question is constructive, and to say very constructive. The idea is what could we do better, right? Rather than just
exactly,
the policy. They have a policy with the Nobel to a maximum of 3 people. So that I imagine that complicates their deliberations, right? In some cases, and then also it ends up, again, seeming to emphasize that what we're celebrating here is some sort of singular and exceptional individual. Now, in the case of the prize in literature, I mean, by definition, it's celebrating an individual, right? Because this celebrates a creative activity that is, in most cases, it's not teamwork, right? I can imagine you can imagine the case of Bob Dylan that was teamwork involved in the sense that he always had musicians around him that probably facilitated his compositions to some degree, but actually he wrote the stuff himself. So, but anyway, now you shift over to science, and it feels, and we celebrate the Einsteins and Feynmans, Marie Curie, and these are historical giants in science, right? And so there's kind of this implication in the public sphere, right, that folks like even me and Gary are sort of like in that same pantheon with those folks, right? And but it's totally missing the point is that we're here because we happen to lead teams that were working on interesting science projects. Well, why were these people there? Because it was interesting to them. They were curious about it. It wasn't like me and Gary were saying, "Hey, we're curious about this. You all come in here, and we're going to tell you what to do." Right? You know,
Mhm.
and your motivation is to please us, right? Totally not right at all. These people, they're only going to come there and do this work, whether it's, Rhonda and Rosalind in my lab, and the folks in Gary's lab, Bruce and um, Reinhardt. Yeah, I'm trying to, I'm, and Amy Pasquinelli, um, those folks were doing that work because they were motivated out of their own curiosity and their own dedication to try to answer questions that were really interesting to them, you know? And so perhaps a missing element would be to, together with honoring, let's say, the PIs, right, to bring in, um, an actual overt recognition and celebration of the teams behind the work, at least the key individuals, let's say, so that they would be celebrated, you know, publicly in some fashion, right? Yeah, because I really like your idea of trying to perhaps shift the Nobel celebration slash discussion um, a little bit so that there would be focus on teams and teamwork. Yep.
Yep. Yeah.
And the very important thing is just what I was trying to say before is that that all this science gets done because the folks who are doing it, which are often students, technical staff, staff scientists, postdoctoral fellows, they are doing creative work in the lab and in their thinking because they are motivated by their curiosity. It's not Ambros and Ruvkin's curiosity 100%, um, and often, the outcome of a project is driven not by the PI, but by the innovations and insights of the folks in the lab collectively, right? And sharing ideas and having discussions where probably nobody in the discussion would be able to trace where the actual final idea came from, right? There's nobody that said, aha, right, and had a brilliant idea. There was a discussion, and then evolving out of that was a really interesting idea that might shape strategies in important ways as they're going forward. So yeah, so it would be really wonderful to be able to, maybe that's what we can do as laureates in our lectures, is maybe bring a little bit more emphasis to this important part of the story. So I really appreciate you bringing this up.
And thank you for also just taking the time and being like helping us, young scientists out with where should we, what kind of questions we should be asking, how should we approach scientific
Yeah,
discovery.
and I can't tell you any of that stuff, right? Yeah, I can't.
You got to figure it out.
Yeah, and I think another question that you had sent me beforehand was about how do people, young people doing science, how to find meaning and purpose in your scientific career.
Yeah, because it's a challenging time for like young scientists with the high living costs, limited job opportunities, and really tough, biotech market in general. And what advice would you offer them, not just about their lab work, but finding meaning and purpose in their scientific careers?
Yeah, this question really hits home because I and all of us, I think, during our journeys in our career, go through times when you question whether or not what you're doing is,
Mm.
is it worth the effort? Is it, um, do I measure up to the task that I'm claiming to to be able to advance? When I write my grants and so on. And so, this, I think that many more of us than we care to admit, perhaps often enough, we don't admit often enough. I think that self doubt is a given and accept and common part of being a human, and and so we all have to, reckon with it and accommodate it and recognize it as being part of human, right? And then also feeling at a given point in your career, especially let's say you're early in your career, and you look forward and you say, okay, what is my trajectory really going to involve before I feel like I've really arrived, you know, where I feel like
Okay.
I've established something that I can be comfortable, feels stable, feels productive, it feels meaningful, and so forth, and I sort of totally sympathize with that. That's also so common. I think it's probably universal, right? And we need to help each other out at those stages. So we need to find communities, micro communities, friends, colleagues, folks who
Yeah.
are to talk about these things, and just share experiences. Folks who are not who not are not stuck in pretending that everything's great and everybody has it all together. I've got it all together, um, you know, find a community who's supportive. Um, and compare notes about how to get through all these things, um, these stages. But I would throw out there that, that whether you're a tech or undergrad working in a lab or a grad student, or a postdoc, you are already a scientist, you're a scientist, you're in the science community, you've you've arrived, right? And you're doing important work, right? And now you might think everybody tells you you're a trainee, you know, and so that makes the tone of that, that sounds like, well, you're not there yet. You're a trainee, right?
Yeah.
But I mean, as you know, we're always a trainee. We're always learning something new. We're always like amateurs, it seems, right? Because the biology takes you places where you're not trained. I mean, who's trained in any of this stuff when you have to do it, right? You train yourself, right? And the point is, you've learned already that, hey, I can learn stuff. I can learn to do stuff. I can find the experts, for example, and ask them, that kind of thing, right? So I feel like everybody should feel, all right, I have arrived. I am a scientist. I'm always a trainee, you know, you know, because if you're a geneticist, right, you follow a mutation, you don't know where you're going to go. I mean, that was the thing with us, right? We followed a mutation. We thought we were studying developmental timing. But it turns out we were, but we also discovered we are studying an interesting kind of gene regulation that's post-transcriptional. It wasn't like, like we wrote a grant, we want to study post-transcriptional gene regulation, so therefore we're going to clone, the gene LET7, or LIN4, right? You go where the biology leads you, so you're always an amateur, you're always a trainee, you always have to find experts, you always have to share your ideas and bounce them off people and get criticism. It's that same process all the time. You got to learn stuff that's out there, read the papers, everything you're doing as a trainee, you just keep doing all your life, and so
That's part of the
oh,
scientific identity. Yeah.
yeah, yeah,
Yeah, amazing. Dr. Ambros, thank you so much for taking the time and having this wonderful conversation with me. I hope we can have many more conversations soon. Massive congratulations again on the Nobel Prize in Medicine and Physiology for
yeah,
2024. And I'll be on campus soon, so hopefully able to see you in person, do some collaborative science as well.
yeah. Stop by when you're on campus. Stop by and say hello. It's always great to
Definitely.
see people in person. That's the other thing I think is very important, is seeing and talking to people in person.
Exactly.
Zoom is great, and it has its advantages, but we don't. When we have the opportunity to see somebody face to face and say hi, we should take it, right? So I love
Definitely. And
part of meeting you.
And likewise, and I saw on the UMass Chan Slack channel, there's always lots of free cookies on the fifth floor. So let's put that as a
Yeah, really?
meeting point.
In the show, really?
Yeah, yeah. I said that's what I've been seeing on the Slack.
Oh, that now I know where to go.
So we can keep this on a little secret, but not anymore. Uh, Thank you so much again, and this was really, really a great pleasure for me.
My pleasure.